This is National Clinical Chemistry Reference Laboratory. Today we will see about the major activities done at this laboratory and the list of major equipment found in this laboratory with their product specification, as well as some of tests or assays done at EPHI, National Clinical Chemistry Reference Laboratory, and their test method or principles, maintenance, calibrator and quality control, preparation of calibrator and quality control, acceptance and rejection criteria, how to check sample quality, result verification, and release of results. When we start from the introduction about Ethiopian Public Health Institute, the Ethiopian Public Health Institute, formerly known as Ethiopian Public Health and Nutrition Research Institute, is re-established as an autonomous federal government institute, having its own legal personality as the main government body in charge of three main tasks, as expressed in the regulation number of 301/2030. One of the major activity is research activity based on national public health research agenda on priority public health and nutrition issues, generate, translate and disseminate scientific and technological knowledge. The second one is surveillance for the early identification and detection of public health risks and prevent public health emergencies through adequate preparedness, alert, timely information during public health emergency, respond effectively and timely, and ensure rapid recovery of the affected population from the impact of public health emergency. The third one is providing referral and backup services and capacity building of health and food science laboratory at a national for quality laboratory services. List of major equipment found at National Clinical Chemistry Reference Laboratory are outlined below. When we see the first one, Cobas E411. Cobas E411 analyzer is a fully automated analyzer that uses electrochemoluminescence technology for immunoassay analysis. When you say electrochemoluminescence technology, it is a combination of three words. There is electrical stimulation, then chemical reaction can take place, luminescence intensity can be produced. It is designed for both quantitative and qualitative in vitro assay determination of broad range of applications, including anemia, bone marker, cardiac, tumor markers, critical care, for example, fertility, hormones, and infectious disease. The analyzer is available as both rack or disk system. The second machine or major instrument found at National Clinical Chemistry Reference Laboratory is Cobas Integra 400 Plus analyzer. This is a perfect solution for laboratories running up to 400 samples per day. It is a broad test menu comprises for over 120 assays and application that consolidate clinical chemistry with specific proteins, therapeutic drug monitoring, and drug abuse testing. This compact tabletop analyzer offers maximum versatility to improve efficiency and reduce costs. It uses the convenient Cobas CPAC reagent format, which standardizes patient results across integrated laboratory networks. The recently received major equipment found at National Clinical Chemistry Reference Laboratories, capillary to flex piercing This capillary flex to piercing or capillary electrophoresis is an instrument utilizing capillary electrophoresis developed to be versatile while providing a clear-cut and precise protein separation with high throughput. This multi-parameter instrument offers a comprehensive menu on serum, urine, whole blood for protein, immunotyping, hemoglobin A1C, hemoglobin, CDT, carbohydrate deficient transferrin testing. The capillaries to flex appearance is equipped with cap piercing technology for whole blood testing, while maximizing the laboratory's workflow. The inversion mixing system provides greater sample homogeneity, resulting in improved accuracy of the results. The other major equipment found at National Clinical Chemistry Reference Laboratory is COBAS 6000. COBAS, when we see the product specification of this COBAS 6000, it has a testing capability, its testing capability is clinical chemistry, ISC, ion selective electrode, hemoglobin A1C, whole blood measurement, and the immunoassay. Throughput is up to 1000 tests per hour combined with photometric and ISC ion selective tests. The samples used on these COBA 6000 machines are serum, most of them are serum, as well as plasma, whole blood, urine, cerebrospinal fluid. And from serum, most of clinical tests, clinical chemistry tests, including ISC and immunoassay. From plasma, ACTH and the D-dimer can be performed. From whole blood, for example, hemoglobin A1C can be done. Urine, from urine to electrolytes, including 24 total protein urine. From cerebrospinal fluid, total protein glucose can be performed. There is automated sample rerun in the dilution mechanism found on this instrument. When we see the test integrity measures used in COBA 6000, there is a contact-free ultrasonic mixing station. There is determination of serum indices. Those are lipemic tests for lipemic, hemolytic, and the icterus. There is a clot and a liquid level detection on this instrument. When you see about reagents, up to 60 reagent cassette positions are available. Automated cassette management system, that is from registration, internal transportation, placement, and disposal. Automatic reagent cassette loading and unloading mechanism also available. This machine has a database, sample database. It can store up to 10,000 data. Some of tests or assays done at EPHI Clinical Chemistry Reference Laboratory are the following. Anemia panel. From anemia panel, for example, ferritin, iron, transferrin, lactate dehydrogenase, folate RBC, and vitamin B12 can be done. The other one is lipid panel. From lipid panel, total cholesterol, HDL, LDL, triglyceride can be done. From bone marker, parathyroid hormone, vitamin D, calcium, phosphate, and alkaline phosphates can be done. From coagulation, antithrombin and the D-dimer can be done. From therapeutic drug monitoring tests, the carbamazepine, lutein, phenobarbital, phenytoin, and valproic acid can be done. Inflammatory markers such as CRP, alpha-1 acid glycoprotein can be done on COBA 6000. Also, fertility panel can be luteinizing hormone, follicle stimulating hormone, prolactin, progesterone, estradiol, and the testosterone can be done with COBA 6000. oncology markers or tumor markers that done in COVAX-6000. For example, for ovarian cancer, CA125, for breast cancer, CA1513, for gastrointestinal tract, CA99, alpha-fetoprotein, for colorectal cancer, and others like acid phosphatase, for prostate cancer, prostate-specific antigen can be done. From cardiac panel, CK total, CKMB, CRP, D-dimer, HDL, LDL, troponin, triglyceride. From thyroid function panel, T3, T4, thyroid stimulating hormone, free T3, free T4, anti-TPO, anti-TG, anti-TSH. From liver function panel, albumin, alpha-fetoprotein, alkaline phosphatase, AST, ALT, bilirubin, phosphatidylglyceride, GGT, LDH. From renal function test, creatinine, urea, cystatin C, uric acid, total protein, potassium can be done in COVAX-6000. From metabolic panel, albumin, total protein, calcium, chloride, magnesium, potassium, sodium, hemoglobin A1C, glucose, insulin, fructosamine, vitamin D, triglyceride, LDL can be done. Other tests like adenocorticotropic hormone, HCG, beta HCG, human chorionic hormone, C-peptide, cortisol, amylase, and lipase can be done. Electrolyte panel, sodium, potassium chloride. During the safe tests, 24-hour urine tests, from CSF, glucose and total protein. Others electrolyte like calcium, potassium, and glucose can be done. Test methods employed on COVAX-6000 are three types. These three test principles or methods of COVAX-6000 are: 1. Absorbance measurement photometry for most clinical chemistry tests. Indirect potentiometric measurement, ion selective electrode. Electrochemoluminescence for immunoassays. Electrochemoluminescence under electrochemoluminescence there is three types of principles. 1. Competitive principle for low molecular weight such as free T3. Second, sandwich principle for large molecular weight such as thyroid stimulating hormone. Bridging principle for antibody measurement like anti-hepatitis B core antigen IgM. When we come to the maintenance, both automatic and manual cleaning is employed. Automatic cleaning by using sodium hydroxide cassette and wash racks containing cleaning solution like sodium hydroxide, IC cleaning solution and activator. Manual by using alcohol and distilled water. When we come to loading of racks, there are color-coded racks used for loading purpose. Sample rack, most of the sample rack are gray rack. Calibrator rack is black rack. Control rack, white rack. Stats for sample that have short turnaround time, red rack is used. Wash rack, green rack is used. When we come to calibrator and quality control, calibrator used both multi-calibrator and specific calibrator can be used for basic chemistry tests. Those calibrators are for example, CEFAS calibrator for automated system, used to calibrate most of the clinical chemistry tests. This is one of the multi-calibrator. When we say specific calibrator, for example, CEFAS, CKMB used to calibrate CKMB, CEFAS lipid for calibration of HDL and LDL, pre-CMAT fructosamine for calibration of fructosamine and others can be used. For immunoassay, the calibrator used is specific calibrator. When we come to quality control, two levels of quality controls are used, both multi-control and specific control can be used. When we see basic chemistry control, basic chemistry, pre-C control, chemical chemistry, multi-1 and multi-2, classified as normal and pathology control can be used. Specific control such as, for example, for fructosamine, pre-C norm fructosamine and the pre-C pass fructosamine, both level 1 and level 2 can be used. Immunoassay control, pre-C control universal for most of immunoassay tests, level 1 and level 2. Pre-C control multi-marker, level 1, level 2. Pre-C control tumor marker, level 1, level 2 can be used. Calibrator and the QC input via specified racks through the input buffer or start port. When we see calibration frequency with every new cassette or lot changes as per stability of the reagent after preventive maintenance. When QC out of range, calibration can be performed. Quality control frequency, quality control can be performed daily after maybe performed after calibration. When we see preparation of calibrator and quality control, calibration can be prepared by adding indicated amount of distilled water using volumetric pipette and letting to sit for 15 minutes and mixing carefully, avoiding the foam formation. Calibrator must be at room temperature before use, and writing the reconstitution date on the bottle is recommended. Control preparation. Control can be prepared by adding the indicated volume of distilled water using volumetric pipette and letting sit for 30 minutes and mixing carefully, avoiding the formation. Before analysis, control must be kept at room temperature before use. Writing the reconstitution date is recommended. Control must be within range. If out of range, repeating the run is recommended. If it is still out of range, investigating the root cause, whether it is due to reagent or calibration or QC preparation. Solving the problem, documenting and resolve the control material and patient samples. Quality control acceptance and rejection criteria. It is based on Westgard multi-rules. This is derived from Six Sigma, which is currently done by EPHI, National Clinical Chemistry Reference Laboratory. For basic chemistry tests and immunoassay, two standard 2 SCD is utilized on COVA 6000. For immunoassay, 3 SCD is utilized. The Westgard multi-rules used in EPHI clinical chemistry are 1 3S, 1 2S, 2 2S, or 4S, and 10X. Those are selected based on the result of 6 Sigma result, and the result of QC is interpreted accordingly. After those samples are analyzed, how to check sample quality? Besides to manual check or visual check, the following serum index for clinical chemistry test is available for quality control or quality check purpose. One is hemolysis index, for example, CKMB, LDH, bilirubin direct, SCTLT, and the potassium tests are highly affected by hemolysis. Having an hemolysis index of 10, 50, 25, 40, 1900 respectively. The other quality check, sample quality check is lipemic index. For example, TRIG, CKMB, ASCT, ALT, those are highly affected by lipemic. Having an index of, for example, for triglyceride 0, for CKMB up to 100, for SCT up to 150, for LDT up to 150 recommended. The other one is icterus or icteric index. For example, bilirubin direct, bilirubin total, fructosamine, cholesterol, creatinine are highly affected by icteric, having an index of 0, 5, 10, 14, 15 respectively. The following, when we come to result verification and result review, after those samples analysis and the quality checks are completed, result verification is done by assigned and qualified laboratory personnel, and those laboratory personnel can check the following: check all panic result which are discordant with previous result, clinically unexpected or mismatch result, and check the quality control, perform dilution when necessary. If the physician requests the test to repeat the serum index level. During result review, check the daily, weekly and monthly maintenance, check the calibration, check the quality control, check the requested result fully performed. If the result need to be verified, check where they were verified well, put your initial. Release of result, check the daily, weekly maintenance assistance, check the quality control, calibration conducted by tested personnel was passed and within acceptable range. Review patient results against analyzer printout. If the instrument is not interfaced, check that the reporting unit matches with the stated standard operating procedure. Check for any flag. Refer the result to the testing personnel if any correction or explanation needed. Before releasing the results, correlate one result to another scientifically, then put your initial and then analyze. Confirm the final result is released on the laboratory information system. When we say something about the quality tests done at Ethiopian Public Health Institute Clinical Chemistry Reference Laboratory, the laboratory is accredited by Ethiopian National Accreditation Office in accordance with the requirements of ISO 15189-2013 general requirements for the competence of medical laboratories. Since INAO is a member of International Laboratory Accreditation Corporation, ILAC, the result performed is accepted everywhere provided that the system essentials are implemented in the laboratory as per the requirements. The instrument available is fully automated, capability to deliver the result within the predetermined TAT, and there is a favorable working environment. This is capillary two-flex sink system or capillary electrophoresis. The capillary two-flex sink system uses the principle of capillary electrophoresis in free solution. With this technique, charged molecules are separated by their electrophoretic mobility in alkaline buffer with a specific pH. Separation also occurs according to the electrolyte pH and the electro-osmotic flow. The capillary two-flex piercing or capillary electrophoresis system has 8 capillaries. The capillary two-flex piercing system, which is currently found at Ethiopian Public Health Institute National Clinical Chemistry, has 8 capillaries functioning in parallel, allowing 8 simultaneous analysis. That is for 8 patients. A sample dilution with buffer is prepared and injected by aspiration at the anodic end of the capillary. A high voltage protein separation is then performed and the direct detection of the proteins is made at the 200 nanometer wavelengths at the cathodic end of the capillary. The capillaries are immediately washed with a wash solution and prepared for the next analysis with buffer. Proteins are detected in the following order. First, gamma globulin, following by beta 2 globulins, beta 1 globulins, alpha 2 globulins, alpha 1 globulins, and albumin, with each zone containing one or more proteins. When we see reagents and materials used for capillary to flex piercing for protein analysis, for example, when you see the buffer is ready to use, and buffer contains pH of 10, around 10. There is also wash solution, dilution segment, and the filters are available. Also distilled water, deionized water, and capicillin, which is used for which contains prolytic enzyme, surfactant, and additives, and the sodium hypochlorite for sample probe cleaning. When we see the procedure for the analysis of serum protein capillary to flex piercings, the capillary to flex piercing system is a multi-parameter instrument for serum protein analysis on 8 parallel capillaries in the following sequence. First, there is a barcode reading for sample tubes for up to 8 tubes and sample racks, followed by sample dilution from primary tubes into dilution segments, and capillary washing takes place, injection of diluted samples, migration under constant voltage controlled by Peltier effect for about 4 minutes. Detection of serum protein by directly scanning at 200 nanometer wavelengths, and electrophoretic profiles appear on the screen of the system. This is the manual steps include placement of sample tubes in sample racks, placement of sample racks on the capillary to flex piercing instrument, removal of sample rack after analysis. Please bear in mind that quality control serum is included with each sequence of protein analysis. Running quality control is a very important, essential component for ensuring the quality of the result. Thus, quality control serum is included with each step of protein analysis. Okay, this is overall about National Clinical Chemistry Reference Laboratories, about the major instruments found in this laboratory, how the quality of results is ensured from sample acceptance to sample analysis as well as sample dispatch. And for the time we have, thank you very much. If you have questions, suggestions, and comments, welcome. And otherwise, I will finish or I will wind up my overview or explanation on this. Thank you very much. Have a nice time. This is overall what activities are done at National Clinical Chemistry Reference Laboratory, about major equipment is found, how quality of test result is improved in the laboratory, and I will conclude my presentation or explanation on this. If you have questions, suggestions, if you have questions, suggestions in the comments, welcome. Unless otherwise, this is I wind up my presentation on this or my explanation on this. Thank you very much. Have a nice time.